Hi everyone, welcome back to class. It's nice to be back here today. To all my returning subscribers, thank you very much for your support so far. And if you are just joining us for the very first time, welcome, welcome, welcome. This is where we had fun to biochemistry. We learn biochemistry in simple and plain English. So welcome, welcome, welcome. Join us. Click the subscribe button, like the video, share the video so that it can reach a lot of students and we can all learn together. So welcome once again. Today's class will be a revision class where we test our general knowledge on biochemistry. But our main focus will be on nucleic acid, nucleotide, nucleosides, the effect of acid and base on the hydrolysis of nucleic acid. That is what our revision will be based on today. So we will check on some questions in these aspects and also provide answers and explanations of why these answers are the correct answers. So let's dive into the revision class proper. Now, the first question is DNA strands are held together by dash. A. Peptide bonds. B. Ionic bonds. C. Hydrogen bonds. D. Ester bond. E. All of the above. DNA strands are what keeps them together. Remember that DNA is a double-stranded molecule and the double strand coiled together. Now, the backbone of the strand are held together by phosphodiester bond between the sugar molecule of one nucleotide and the phosphate molecule of another nucleotide. While the nitrogenous bases of each strand project inward. So now the bond between these nitrogenous bases are hydrogen bonds, and these are the bonds that also hold the molecule, the DNA molecule, together as a whole. So the answer there is C, which is hydrogen bond. The next question is pyrimidine bases include dash. Pyrimidine bases include dash. A. Adenine and guanine. B. Guanine and cytosine. C. Cytosine, thymine and uracil. D. Adenine and uracil. E. None of the above. Now, remember in our purine and pyrimidines, where we under the nitrogenous basis, we said the purines are the adenine and the guanine, while the pyrimidines are the cytosine, uracil, and thymine. So in this question, the pyrimidines that are there are C, the cytosine, thymine, and uracil. Those are the pyrimidine bases. The next question, the process of synthesizing DNA from DNA is called dash. The process of synthesizing DNA from DNA is called dash. A. Translation. B. Replication. C. Transcription. D. Mutation. E. None of the above. The process is replication. You just replicated DNA from DNA. That is replication. But if you are moving from DNA to RNA, that is transcription. If you are moving from DNA to DNA, that is replication. If you are moving from DNA to RNA, that is transcription. So the answer to that question is B, replication. Let's move on to the next question. A nucleotide consists of dash. A nucleotide consists of dash. A. Base, sugar, and phosphates. B. Base, sugar. C. Base, phosphate. D. Base only. E. Phosphate only. Nucleotide. The main difference between nucleoside and nucleotide is that nucleoside consists of only nitrogenous base and ribose sugar, but nucleotide consists of nitrogenous base, ribose sugar, and a phosphate group. So phosphate, the presence and the absence of phosphate group is the main difference between nucleotide and nucleoside. Now, for this question, a nucleotide consists of dash. The answer is A, which is base, sugar, and phosphate group. The next question, the backbone of DNA consists of dash. A, sugar and phosphate. B, sugar and base. C, protein and phosphate. D, lipid and sugar. E, sugar only. Now, remember our DNA structure. We said DNA is a double helix, and that double helix structure as they are joined together by hydrogen bond. The double helix is joined together by hydrogen bond, but on the backbone we have the phosphate group and the sugar molecule, which are joined together by phosphodiester bond. So now, to answer this question, the backbone of DNA consists of dash. The answer is A. That is a sugar molecule and a phosphate group. That is the two things that come together to form a phosphodiester bond. Next question now. Nucleic acids are polymers of dash. Nucleic acids are polymers of dash. A, amino acids. B, fatty acids. C, nucleotide. D, nucleosides. E, monosaccharides. Meaning that what makes up nucleic acid? Nucleic acids are polymers of nucleotides. Nucleotides are the ones that join together that make nucleic acid. And what actually joins together to make nucleotides in the first place? We have our phosphate group, we have our sugar molecule, and we also have our nitrogenous base. So the answer to that question is C, nucleotides. The next question, DNA possesses an overall negative charge because of its dash. DNA possesses an overall negative charge because of its dash. A, phosphate group. B, nitrogenous bases. C, hydrogen bond. D, ribose sugar. E, deoxyribose sugar. Now, what actually makes a DNA molecule to be negative charged is the presence of that phosphate group, which is at the backbone of that structure. So the presence of that phosphate group is the one that provides the negative charge to that molecule. So the answer there is A, that's phosphate group. Now, the next question, DNA denaturation involves dash. DNA denaturation involves dash. A, hydrolysis of phosphodiester bond. B, destruction of bases. C, formation of nucleotide. D, separation of the two strands. E, joining of the two strands. Denaturation is just in another way you separate DNA. Remember our DNA is a double-stranded structure. Now, for you to denature your DNA, you mean that you want to separate the strand. You can denature making use of chemical, you can denature making use of heat. So, if you now denature, the result that you get from there is a separate strand. So when you denature, you have two separate strands. So the answer to that question is D, separation of the two strands. Now, the next question, the two major types of nucleic acids are dash. The two major types of nucleic acids are dash. A, RNA and DNA. B, DNA and ATP. C, RNA and ADP. D, RNA and protein. E, none of the above. Remember our nucleic acid. We said our nucleic acid help us to store information, transmit information, and also help in the expression of this information. So, and it is made up of two main things, which are the DNA and the RNA. The DNA are the ones that are in charge of storing genetic information, while the RNA are in charge of transmitting this information and also eventually leading to protein synthesis. So the answer to that question is A, DNA and RNA. Those are the two main types of nucleic acid that we have. The next question, a nucleoside consists of dash. A, sugar and phosphate. B, base and sugar. C, base and phosphate, D, sugar only, E, phosphate only. A nucleoside. The main difference between nucleoside and nucleotide, as I said earlier, is the presence and absence of a phosphate group. So a nucleoside only has a sugar molecule and nitrogenous base. It does not have a phosphate group. So the answer to that question is B, base and sugar. The next question now. DNA is preserved as the genetic material because it is dash. A, single-stranded, B, rich in uracil, C, easily hydrolyzed, D, more chemically stable than RNA, E, none of the above. Now, let's take them one after the other. Is our DNA single-stranded? No. Our DNA is double-stranded. Is our DNA rich in uracil? No. Uracil is only present in RNA. Now, is our DNA easily hydrolyzed? The answer is also no. It depends on the solvent that is present. Is it acidic solvent? Is it basic solvent? So it depends. That will depend on what we hydrolyze it. Fourth one is more chemically stable than RNA. Yes, DNA are actually more stable than RNA. They are very, very stable than RNA. So that makes them favorable and that makes them good in storage of genetic information. So the answer to that question is D, more chemically stable than RNA. The next question, ATP is known as dash. ATP is known as dash. A, genetic code, B, energy currency of the cell, C, structural protein, D, hormone, E, none of the above. ATP, adenosine triphosphate. That is the only language that the cell understand in terms of exchange. So we can say that although the cell doesn't recognize this naira, they don't spend dollars, they don't spend pounds, they don't spend all this currency, but the only one that they recognize and they spend is ATP. So that's the only energy currency of the cell. So the answer to that question is B, energy currency of the cell. So when you hear these biochemical terms where people are saying the energy currency of the cell, the currency, this, that, what they are referring to is ATP. The next question, DNA is generally dash. A, single-stranded, B, double-stranded, C, triple-stranded, D, circular-stranded, E, not-stranded. The answer there is B, double-stranded. Our DNA is double-stranded molecule. So our DNA comprises of two polynucleotide chains that wind or coil around each other. That is why we call it double helix structure. So the answer to that question is B, double-stranded. The next question, hydrolysis of nucleic acid involves dash. Hydrolysis of nucleic acid involves dash. A, addition of water, B, removal of water, C, oxidation, D, reduction, E, none of the above. Let's break it down. Hydrolysis. Hydro, what does it mean? Water. Lysis, what does it mean? Breakdown. So when we now join them together, that means we want to break down something in presence of water. So just know that, okay, you are breaking down something and water is also there. So just know that water is involved. Now, the options are now that, is it that you add water or you remove water, or is it oxidation or reduction? The answer is A, addition of water. So hydrolysis now, hydrolysis of nucleic acid involves addition of water. Now, the water that you now want to add, it now depends. That water you are now adding, it now depends. Okay, is it acidic? Is it basic? Is it alkaline? So that one will now determine what will happen to the nucleic acid in question. Now, so the answer to that is A, addition of water. Now, the next question, dash hydrogen bond are formed between guanine and cytosine. Dash hydrogen bond are found between guanine and cytosine. A 1, B 2, C 3, D 4, E 5. How many hydrogen bonds are between cytosine and guanine? Remember, your adenine and thymine had 2 hydrogen bonds. And so your cytosine and guanine have 3 hydrogen bonds. So the answer to that question is C, 3. So 3 hydrogen bonds are always between your cytosine and your guanine. Now, the next question, DNA stands for dash. DNA stands for dash. A, deoxyribose nitrogen acid. B, deoxyribonucleic acid. C, deoxyribonucleic acid. D, deoxyphosphate acid. E, none of the above. The correct answer there is B, deoxyribonucleic acid. DEoxyribo nucleic acid. So B there is the correct answer. The next question, the sugar present in RNA is dash. What is the name of the sugar molecule that is present in the RNA? Now, the first, the A is the deoxyribose sugar, B ribose sugar, C glucose, D fructose, E none of the above. The answer there is B, ribose sugar. That is where the name was coined out from, ribonucleic acid. It's a 5 carbon sugar, so it's the ribose sugar that is present in RNA.